In this recorded lecture, we're going to review photosynthesis, and we're going to review the light reactions of photosynthesis, but we're going to focus more on the Calvin cycle reactions. So these slides are from chapter 7, and what you're looking at is a chloroplast, which is the organelle that carries out photosynthesis. And just to remind you, the light reactions of photosynthesis occur in the thylakoids. You're looking at a stack of thylakoids here. The stack is called a granum. The individual green discs are called thylakoids. The light reactions are also responsible for splitting water and producing oxygen gas. And the light reactions absorb solar energy or sunlight and produce NADPH and ATP. Then the Calvin cycle reactions convert the NADPH and ATP, or use the energy from, the Calvin cycle reactions use the energy from NADPH and ATP to convert carbon dioxide into glucose or some other carbohydrate. And these Calvin cycle reactions occur in the stroma of the chloroplast, which is the fluid that surrounds the thylakoids. Okay, so now we're going to move on to, we're going to review the non-cyclic electron pathway for the light reactions. So there's a non-cyclic pathway for the light reactions, and then there's a cyclic pathway. The non-cyclic pathway begins with photosystem 2, and everything that we're looking at here on this page, this illustration with photosystem 2 and photosystem 1, and these electron acceptor molecules, these are all part of the thylakoid membrane. So the non-cyclic pathway begins with photosystem 2. Photosystem 2 is a pigment complex that absorbs sunlight energy or solar energy and excites an electron from the reaction center. And this reaction center is actually a chlorophyll A molecule, a special chlorophyll A molecule, and it's only shown in red just so that it will stand out. It's actually green because all chlorophyll molecules are green pigments. But the electron that is excited from the reaction center chlorophyll A molecule is transferred or transported to an electron acceptor molecule in the thylakoid membrane. And from there, it is passed down an electron transport chain, which is a series of protein carrier molecules in the thylakoid membrane that pass these electrons along. Using the energy from the flow of electrons, hydrogen ions are pumped across the membrane, and then those hydrogen ions are used to generate ATP. So photosystem 2 generates ATP, and photosystem 2 is also responsible for splitting water to replace the electrons that are lost from the reaction center chlorophyll A. So when water is split, oxygen gas is released, and that is the part of photosynthesis where the plant releases oxygen gas to the atmosphere. Now, the electrons flowing from the electron transport chain eventually arrive at photosystem 1. And then photosystem 1, another pigment complex, absorbs solar energy and excites the electron from the reaction center chlorophyll A molecule. In this way, it's very similar to photosystem 2. But when it excites the electron, and that electron eventually is transported to the electron acceptor on the thylakoid membrane, those electrons from photosystem 1 end up going on to form NADPH. So photosystem 1 forms NADPH, photosystem 2 forms ATP and splits water. So those are the differences in the two photosystems. Now, the ATP and the NADPH are the products of the non-cyclic pathway of the light reactions. They go on to power the Calvin cycle reactions, where carbon dioxide is converted into a carbohydrate, such as glucose. In your notes, you wrote down that photosystem 2 is a pigment complex and electron acceptor molecules that split water and release oxygen and also produce ATP. The photosystem 2 leads to the electron transport chain, and the energy from those electrons pumps hydrogen ions into the thylakoid space inside of the thylakoid disc. And then those hydrogen ions eventually flow down their concentration gradient through this ATP synthase complex, which is actually an enzymatic protein as well as a channel protein that is, its role is to produce ATP using the concentration gradient of the hydrogen ions. So basically you have photosystem 2, which leads to the electron transport chain, which leads to the ATP synthase complex and the production of ATP. Then the electron transport chain takes electrons to photosystem 1, and photosystem 1 generates NADPH. Both of these, ATP and NADPH, are used in the Calvin cycle reactions. Now, that was the non-cyclic pathway. Remember, the non-cyclic pathway contains photosystems 2 and photosystem 1. The cyclic pathway only contains photosystem 1, and in the cyclic pathway, photosystem 1 is used to absorb sunlight energy, excite electrons, and then an electron transport chain leads to the generation of ATP. But it only, in the cyclic pathway, you only have photosystem 1 and you only have the production of ATP. Just don't get confused, because in the non-cyclic pathway, ATP is produced from photosystem 2. Okay? So in the cyclic pathway, you only have photosystem 1 and you only have the production of ATP from the electron transport chain. And that ATP is then used by the Calvin cycle. Now, what we're looking at here is a thylakoid membrane. Notice it is green. Here's our thylakoid membrane. And we have all of these large orange molecules that are proteins within the membrane proteins, kind of like the plasma membrane proteins, only these are found in the thylakoid membrane. And we have some pink molecules that serve as electron carriers. So let's start with the electron transport chain. Okay, so, or actually let's start with photosystem 2. Photosystem 2, pigment molecules are right here. And those pigment molecules absorb sunlight energy, excite an electron, and that electron is transported to this large orange electron acceptor. And then the electron travels and it's carried from this little molecule called PQ, and then this electron carrier here, another electron carrier, all the way to photosystem 1. Okay, so this is the electron transport chain portion. You have photosystem 2, electron transport chain, and the flow of electrons here is used to pump hydrogen into the thylakoid. Hydrogen ions build up in the thylakoid. And then those hydrogen ions encounter ATP synthase and flow down the channel, down their concentration gradient from where their concentration is high to where it is low. And then the flow of hydrogen ions is used to generate ATP. So we've got photosystem 2, electron transport chain, ATP. Now, the electron from the electron transport chain is still there, so it has to go somewhere. So it enters photosystem 1. More sunlight energy is absorbed, and it excites that electron, and that electron ends up being used to produce NADPH. So now we have our two products of the light reactions, ATP and NADPH. Now they're going to go on to the Calvin cycle. And the Calvin cycle reactions is a cyclical series of reactions that takes atmospheric CO2 or carbon dioxide and converts it into carbohydrates or food. There are 3 stages in the Calvin cycle. There's carbon dioxide fixation, carbon dioxide reduction, and RUBP regeneration. In the first step, the fixation of carbon dioxide is attached to a molecule called ribulose biphosphate, RUBP. Carbon dioxide has 1 carbon in it, and then RUBP has 5 carbons. So when they're attached, you have a 6-carbon molecule that is immediately split into 2 3-carbon molecules. But the most important thing is that carbon dioxide is attached to RUBP. The enzyme that carries out this reaction is called RUBP carboxylase, or it's also known as Rubisco. This enzyme is the most abundant enzyme on the entire Earth. So that just lets you know how important photosynthesis is. This particular enzyme, if we were to count up the number of molecules of enzyme on the whole Earth, Rubisco is by far the one that we would count the most of. It is the most abundant enzyme on the Earth. So just again, it's just one way to let you know how important photosynthesis and the Calvin cycle are. Because plants provide us with oxygen and they provide us with food. Now, this is an illustration of the Calvin cycle reactions. If you look at this portion here and kind of like in the upper left, you see where RUBP is attached to carbon dioxide. This is the step 1. It's the fixation of carbon dioxide to RUBP. And after step 1, you have these molecules called 3-PG. Doesn't really matter what it means, but I'll just say it, 3-phosphoglycerate. And that 3-phosphoglycerate is converted to BPG, and then our final product of the Calvin cycle, which is G3P. G3P stands for glyceraldehyde 3-phosphate. So G3P can be converted to carbohydrates like glucose. Okay, so first step is the fixation of carbon dioxide to RUBP. Second step is the production of G3P, and we call that carbon dioxide reduction. So this is the second step, and it ends with the production of G3P. Okay, carbon dioxide reduction produces G3P. That is our glyceraldehyde 3-phosphate, which is our main product of the Calvin cycle. That can go on and be converted to glucose or other carbohydrates. Now, for every 6 G3Ps that are produced, 1 can be used to make glucose, and 5 are used to regenerate RUBP so that the cycle can continue. So for every 6, only 1 can be used. 5 go back into, they're recycled. They go back into the Calvin cycle and are used to regenerate RUBP. So we have those 3 steps. We have step 1 is attaching carbon dioxide to RUBP by the enzyme Rubisco or RUBP carboxylase. That is the fixation of carbon dioxide step. The second step is the reduction of carbon dioxide with the final product being G3P. That is a 3-carbon molecule. So carbon dioxide is reduced to G3P. And in step 3, 5 of those G3Ps are used to make 3 RUBPs. And that is because the G3Ps have 3 carbons, so 5 G3Ps with 3 carbons each equals 15 carbons. 3 RUBPs having 5 carbons each would account for those 15 carbons. So you just have to do the math. You need 5 G3Ps that have 3 carbons each to regenerate 3 of the 5-carbon RUBPs.